The cerebellum, Latin for little brain, is a key component of our central nervous system. Located in the ventral posterior area of the skull, the cerebellum is a major structure of the hindbrain. It's responsible for balance, posture, coordination, and a number of other motor skills. It contains up to 50% of our brain's neurons, and cerebellar injury can cause severe cognitive impairments. But how is this essential part of our brain created in a growing embryo? In this video, we'll study the development of the cerebellum from Rhombomere 1. Well before the existence of the cerebellum, a developing embryo's early nervous system is called its neural tube. Within a few days of fertilization, the hindbrain region of this neural tube develops several small transient segments that we call rhombomeres. We number these rhombomeres from the anterior end to the posterior end, and there are typically between 7 and 9 in total, depending on the species. Rhombomeres 2 and higher give rise to many important nerves, including the vagus nerve, glossopharyngeal nerve, abducens nerve, facial nerve, and trigeminal nerve. But Rhombomere 1, or R1, will give rise to the cerebellum. But before R1 can make this transformation, it requires signaling from the isthmic organizer at the midbrain-hindbrain boundary. Once the isthmic organizer is established, by antagonistic expression of the homeobox genes OTX2 and GBX2, it then begins secreting a substance called fibroblast growth factor 8, or FGF8. This FGF8 molecule is a key orchestrator for multiple stages of cerebellar development, regulating both gene expression and cell survival. In 2009, Sato and Joyner showed that both the strength and duration of FGF8 signaling must be finely tuned to properly induce cerebellar development. In fact, this proper expression of FGF8 is so important that a slight dip in its signaling can cause the loss of the posterior midbrain and vermis, while a complete loss of FGF8 can even prevent the entire midbrain and cerebellum from forming. However, if FGF8 is accurately expressed, it will induce continuous morphogenesis in Rhombomere 1 that, over time, will transform the rhombomere into the cerebellum. FGF8 doesn't work alone, though. The FGF8 gene also coordinates with a number of other known genes, such as the homeobox genes EN1 and EN2, and the paired box genes PAX2 and PAX5. These genes are believed to have complex interlocking interactions, with some of them working to induce FGF8 expression, and others helping regulate and maintain FGF8 levels. Recent findings also suggest that there may be additional upstream signaling molecules involved in this pathway during the establishment of the isthmic organizer. The system may be complex, but if all genes are properly expressed at the right times, they should allow Rhombomere 1 to fully develop into the cerebellum. This transformation is not only essential for proper cognitive function, but is also a pristine example of the beautiful intricacy of our developing nervous system.